All right, well, welcome everyone to this week's edition of the EV Journal Club. Thank you all for joining. And I'd like to thank Andy Chen in particular, who's a research scholar at Cedars-Sinai in the lab of Dolores DeVizio. And so Andy had reached out to me a couple weeks ago and mentioned that he was going to be presenting this paper at his local journal club. And so he kindly also agreed to present to us today. So thank you, Andy. And I think we're all looking forward to hearing more about this resource paper and what it has to offer the community. So Andy, I will hand it over to you.
Okay, so hello. Once again, I'm Andy Chen. I work with Dolores DeVizio. And today I'll be presenting this paper titled Extracellular Vesicle and Particle Biomarkers Define Multiple Human Cancers. So once again, this is a resource article. It's not a research article. It was published by the David Lighting Group. And this is probably the most comprehensive and largest proteomics study that has ever been done on extracellular vesicles. But once again, it's a resource article. So it's more about providing a potentially useful tool for your research rather than providing novel insights or new discoveries. But there are some interesting findings that they had as well. So one of the things that they were trying to address with this resource paper is that one of the big reasons a lot of people are working on extracellular vesicles is that they are potential sources of cancer biomarkers that can be found from biofluids in the, say, from biofluids such as serum or plasma. But we really don't know common proteins that are, one, enriched in cancer versus non-cancer, or just enriched in EVs from various different types of cells, period. So they were trying to do this comprehensive proteomics analysis of the proteins in these EVs to try and address these basic questions that have been left unanswered for years. So first, let's try going forward. So what are they looking at in this paper? So they isolated their extracellular vesicles, which they call EVPs. Okay, so this is their isolation protocol here on the left. So they're using a very standard ultracentrifugation protocol for the isolation of small extracellular vesicles. This includes the 3 populations that the Lighting Group has shown to be present in this 100,000 times G pellet, including 2 different populations of exosomes, XOS and XOL, which are different from each other by the migration speed through density gradients. They have slightly different sizes as well. It also includes very small extracellular particles called exomeres, which lack the lipid bilayer. And we still really don't know what they are, but they're like roughly 50 nanometers or smaller. So basically they're calling their preps EVPs because they contain these small EVs as well as non-vesicular exomeres. So nanoparticle tracking analysis shows that the size of the particles is roughly peaks at around 100 nanometers, and that transmission electron microscopy, you see images of what appears of particles that have vesicular morphology, which gives them confidence that they're studying what they think they are studying. So what did they do with these samples? So they collected a huge number of samples from both human and mouse, which will come from for both tumor and non-tumor samples. And so this is the samples that they'll be using throughout the entire paper. Just a note that the tumor samples are heavily enriched in lung and pancreatic cancer, but there are several other types of cancer in there as well. So the first thing they wanted to look at is the correlation of their different types of samples that they collected with each other. Interestingly, they found that although the cell lines and the tissue explants have very high correlation with each other, when you looked at the correlation of these samples with plasma samples and biofluids, there was very poor correlation between the cell lines and tissue explants with plasma and serum. In contrast, the plasma and serum samples had a much higher correlation with other biofluids such as bone marrow, lymphatic fluid, and bile duct fluid, which suggests that the extracellular vesicles coming from the plasma and serum, most of them might not originate from the primary tumor, if you're thinking about cancer patients. While they might come from other cells, such as the immune cells that are circulating in the patient. So another thing that they wanted to highlight is that when you look at human versus mouse cell lines and human versus mouse tissue explants, there's a very high correlation. However, the correlation between human plasma and mouse plasma was significantly weaker. It's still decently strong, I think, but it's not as strong as the correlation that you see within cell lines. And so their claim was that the mouse plasma might not be a good study model for studying human plasma, but the mouse cell lines and tissue explants might be better at recapitulating what's happening in the human. So one of the most interesting things that they found in this paper is that they looked at the classical small EV and extramural markers in the different samples. Interestingly, they found that all these classical EVP markers, although they were heavily abundant in the cell lines and tissue explant samples, when you looked at the plasma and serum samples, most of these proteins were heavily depleted, with only two really being commonly abundant, which is HSPA8 and CD9. With the rest of them, they were pretty much absent in plasma and serum, suggesting that the small EV markers that we identified that are commonly used in research that were identified from cell lines might not be applicable to all biofluids. So to address this, they tried to find common small EV EVP markers. And this is the list of new small EVP markers that they found. Some of these proteins are very familiar, such as fibronectin and beta-actin, whereas other of them have been less well studied in extracellular vesicles. But the key thing about all these EVP markers is that you can find them consistently across every different type of sample type that they studied, including cell lines and plasma and serum. So next, they wanted to validate whether they could actually find these new pan-EVP markers in various different cell lines and tissue explants by two different methods. They use Western blot and ExoView. So ExoView is an immunoaffinity technology that looks at, that's typically used for the quantification of exosomes. An antibody is used to immobilize the EVs onto a chip, and then there's fluorescence imaging is used to detect the presence of vesicles. So they were able to find most of these new pan-EVP proteins and cell lines. Some of them were detectable by tissue explants. Interestingly, if you look at all the different types of samples they have, they see a pretty large variety in the EVP proteins that are detectable in tissue explants, suggesting that there might be more heterogeneity in the tissue explants than even the plasma samples, which is very surprising. Some of these EVP proteins can be detected in all the tissue explants, such as stomatatin, tolasostat actin, and beta-2 microglobin. And several of these proteins were present. It could be also identified in EVPs isolated from human plasma. What's interesting is that they did not validate the lack of the inability to detect the common EVP proteins such as TSG101 and CD81 by mentioning those two in particular because they do have TSG101 and CD81 in the cell lines and tissue explants, but they're not showing the Western blot from plasma, which is very curious because it would be nice to see some validation for this lack of discovery of these two markers that they showed here in the plasma samples. So then they could also find two of these EVP proteins on the surface of extracellular vesicles, myosin and beta-2 microglobin, suggesting that they might be good for immunoaffinity. However, one of the downsides of these two proteins that I'm seeing is that there's a very large sample variability between these two molecules, as opposed to CD9, CD81, and CD63, which are much more heavily used and have a much more narrow standard deviation between the three different biological replicates. So this does suggest that you can find that this suggests that there's more work that needs to be done to identify pan EVP markers and validate them for further use, but suggests that some of these proteins are promising candidates. So what they next wanted to do is they wanted to try to use their proteomics data to see if they could identify cancer-specific EVP proteins. And the way they did this is they took tissue explants from mainly two different types of cancer, pancreatic adenocarcinoma and lung adenocarcinoma. These are the two most commonly used cancer types in their data set. And they took tumor tissue and non-tumor adjacent tissue, and they cultured them ex vivo. And then they isolated extracellular vesicles from these ex vivo cultured tissue explants, and then did proteomics on these isolated EVPs. And so what they found is that they could find enriched and specific, I'm only showing the enriched proteins here, but they could find both enriched and specific proteins to both pancreatic cancer and lung cancer. And gene ontology analysis of these enriched proteins showed that the pancreatic cancer tumors were enriched in gene ontologies related to the EMT, the cytoskeleton, the filament assembly, and the extracellular matrix, whereas in contrast, the lung cancer specific or enriched proteins were enriched for proteins that are involved in mixed signaling and RNA processing, which gave them some evidence that the different types of tumors have different signaling pathways that are being activated. So you might be able to use proteomics to define which type of tumor a patient has. And that's something they will go into more detail in the future, in later slides. So another one of the things they wanted to look at is, since the immune modulation is one of the most commonly studied pathways regarding to the interaction between extracellular vesicles and cancer cells, they wanted to see if they could find some immune modulatory proteins, and they looked specifically at damage-associated molecular pattern proteins, which are known to modulate the immune system, that were released by damaged or dying cells and are known to activate the innate immune system. Not surprisingly, they found several molecules, DAM molecules, were specifically induced in the tumor tissue as opposed to the adjacent tumor tissue. However, in pancreatic tumor EVPs. However, there are surprisingly several proteins that were active in both the adjacent tissue and the tumor tissue itself. In contrast, for the lung tumor EVPs, they did not find any proteins that were highly specific to the tumor tissue, rather all the DAMP proteins that they found in the EVPs were present both at the in the non-tumor adjacent tissue as well as in the tumor tissue, and also present in distant tissues as well, suggesting that this activation of DAMP proteins in the extracellular vesicles is coming from a systemic response to the cancer. It's not coming directly from the cancer cell itself. So they next want to see if they can create a list of proteins that could be used to predict the presence or absence of cancer in cancer patients. And they started with tissue explants. So they used random forest classification of a variety of different tumor and non-tumor samples. Once again, the tumor samples were heavily enriched in lung and pancreatic cancers. And then they compared the abundance of these proteins to proteins enriched from non-tumor or isolated from non-tumor tissue EVPs, including, once again, pancreas and lung, which make up the bulk of the non-tumor tissues, but also bone marrow makes up a considerable portion as well. So yeah, using this random forest classification scheme, they came up with this list of proteins which could be used to predict with a sensitivity of 90% and a specificity of 94%, the presence or absence of cancer. However, one of the problems with this method is that they're using tissue explants. If you're doing tissue explants, you already know at that point that your patient has the person has cancer or not. So the next step is to go and look and see if you can do the same thing from plasma or serum. So when they were isolating EVPs from plasma or serum, when they were analyzing the data, they found an interesting result in which, although some of the prostate cancer explant EVs were found in the plasma, there were several EVs from the pancreatic cancer plasma which could not be found in either the pancreatic cancer cell explants or adjacent tissues, suggesting that these proteins are coming from other sources of the body, such as possibly immune cells or other organs. So once again, they used random forest classification. They wanted to also validate the specificity of the mass spec. So they used targeted mass spec to see if they could find some of the proteins by more sensitive methods, such as targeted mass spec and ELISA. They were able to validate some of these proteins by both of these methods. So once again, they were able to identify a panel of proteins that could predict the presence or absence of cancer from tissue samples, but then they wanted to look at whether they could do the same for plasma EVPs, and they were able to generate a much larger list of 47 proteins that were able to predict the presence or absence of cancer from plasma EVPs with a sensitivity of 95% and a specificity of 90%, suggesting that you can potentially use plasma EVP proteins to predict the presence of cancer in patients. And so the last thing they wanted to do is, so if say you have cancer, based on this will tell you whether you have, the signature will tell you whether you have cancer or not. Can you use this proteomics to tell you what type of cancer you have? So they basically did t-SNE analysis on extracellular EVPs isolated from various different cancer cell types, and they found that they were able to, they did clustering analysis, and they found that the cancers from different types of cancer types clustered differently, separately from each other. So one of the things that's missing from this study, to me, is that I feel like they really need to have the healthy patients here to make sure that the healthy patients cluster separately from the different types of cancer patients that they have. But otherwise, they're suggesting that if you know you have cancer, you can potentially use proteomics to tell you what type of cancer they have. But this also needs to be done on a much larger subset, because there's also there's a large degree of heterogeneity and many different drivers for each type of cancer. So it still needs to be determined whether this still holds up when you're looking at a larger, more diverse set of each type of cancer. So in summary, what they've done is they've looked at extracellular vesicles from a variety of different sources, peripheral blood, such as plasma and serum, adjacent tissues, tumor tissues, and distant organs. And they found that the proteomics, they compiled a huge data set of proteomics, which could be useful for your studies in the future. And they suggest that you might be able to use the proteomics from plasma and serum to predict the presence and maybe even the type of cancer in the body. They also, probably the biggest finding they found is the importance of identifying pan-EVP markers that include EVPs present in plasma. They've listed a few proteins which might serve as novel markers for plasma and serum, as well as other tissue EVPs, but they still need to be validated. And so, yeah, thank you for your attention. Are there any questions?
Right, thanks very much, Andy, for that very clear presentation. I think there's a lot of material here that people can look into, and I guess I want to start with a question, not just to you, but basically everybody. Did you see any proteins in here in these figures that were a surprise to you? I mean, quite apart from the the absence of some of the markers that we traditionally have used for EVs. And I'm going to, just a minute here, I have to allow people to unmute themselves now. So if you have, I'm going to go through the questions that have been put in the chat box, but if you have additional comments, I'll call on you and you can unmute yourselves.
Yeah, regarding your question, I thought most of the proteins that I saw that I recognized were very familiar or already familiar with me. I did not see something that I was not expecting to see at all. There was a high enrichment of integrins and extracellular matrix proteins, which are highly enriched in previous studies. But yeah, I did not see anything that I was not expecting to see, or that I would not have expected to see.
So it looks, I mean, from Roger Alexander had asked about centrifugation. And so I think that, this paper is a good example of how, I think that the authors were very careful about the terminology that they use. So they're not suggesting here that they're just looking at EVs.
That's correct. I mean, they are doing just ultracentrifugation. There is a risk of having non-EV proteins that are co-precipitated with those samples. So that is definitely a risk that they have in those samples.
Yeah, and so that's that's why they're using that term EVP, or extracellular vesicles and extracellular particles, because they don't want to try to make the case that they're just looking at EVs. I think that's a safe way to go. But then on the other hand, I think they're probably looking at a pretty broad population. Okay, so let's go to Kashipra Kapoor. Oh, actually, I guess this also has to do with ultracentrifugation. So there was not any additional methods that they used. Is that correct?
That is correct. It was just simply ultracentrifugation, so that they know they're isolating a diverse population of extracellular vesicles and extracellular particles here, but they don't go further to do, say, density gradients or anything else.
Mm-hmm. And another question then here from Paula. Paula, do you want to ask your question?
So I was just thinking it might be useful, in addition to having normal samples for comparison, like you pointed out, to make sure they do separate away from the cancer samples, maybe samples from inflammatory diseases that, they may also have signatures.
Yeah, definitely. I totally agree with you. It would definitely help to see if what you're saying is just a signature of, say, chronic fibrosis rather than cancer per se, or something like that. So yeah, if you want to say that, if your goal is to identify cancer-specific proteins, you definitely need more other systemic diseases that could potentially alter the EVs as well.
And then John Sperduto, did you want to follow up on the question that you asked, or has that been addressed? I mean, I think it's pretty clear that they do have other particles in here, so this is not just about EVs. But did you want to follow up on that?
No, more or less addressed it. Granted, particles could mean a vast array of things you're delving into. Now it could be tumor secreted factors that have aggregated. But no, yeah, that addressed my question. Thank you.
And then Genevieve.
Kind of a stupid question, but I remember a few years back, there was a publication from Sonia Mello that blood exosomes from pancreatic patients were enriched in glypican-1. So I was just curious whether glypican-1 had actually appeared as a marker in this study.
You were talking about integrin beta-1, is that, sorry, did I get that correct?
Glypican-1.
Oh, glypican-1. I had to go back and check to see whether I saw glypican-1 in these
I
patients.
kind of tried to see in the figures that you showed, but I missed it. I was just curious.
Yeah, I had to go back and check. I'm not seeing it right now. Let me try sharing my screen again. So these are the highly enriched proteins in pancreatic and lung tumors. And at a glance, I'm not seeing it, but it could also be in this. They have several huge lists proteins here. So there's definitely lots of data. If you have a specific question, there's lots of data you can go through and look at it.
No, I thought that was kind of that would have been discussed since it had been published that it was a biomarker.
Yeah, I'm not, well, in this particular figure, I'm not seeing it. It could also be in one of the other figures that are looking at, well, they also have similar figures to this. This is the highly enriched proteins in pancreatic and lung. They also have the specific proteins to pancreatic and lung. It could be in that figure as well.
Yeah, I guess there has been some additional evidence that's come out that, the glypican-1 can be associated with pancreatic, but then it's also found in some other cancers. Our next question then is from Susan about serum and plasma. Susan?
Yeah, I was trying to quickly look at their data on the plasma and serum, and that was pretty concordant, but not perfectly so. And it didn't look like, it looked like there were some things in plasma that weren't in serum, which I sort of expected, but it looked like there were some things in serum that weren't in plasma, or were, higher in serum versus plasma. Did they quantify, I mean, how should I think about their quantification, I guess, is my question. So there were these red boxes that you had with serum versus plasma. And so I'm just trying to, a lot of the biobanks that I deal with is serum. And so I've been trying to figure out serum versus plasma.
Yeah, they saw a lot. The red boxes was the abundance, and let me get my screen again. You're talking about this.
Yep.
So basically, the red boxes, out of all the samples that they looked at, could they detect, like, what percentage of these samples could they
Actually,
detect?
it was the slide after that.
This one.
That one, I think. So plasma was 120, serum. Yeah, exactly. And
Yeah,
so,
so
yeah,
so there's a lot of similarity that's shown here between plasma and serum. The numbers here just represent in what percentage of those samples could they confidently detect these proteins.
I see. So it's not quantitative,
This is not a quantification.
Yeah.
There could be huge differences in, say, the abundance of fibronectin 1, which is the top one, and different in plasma versus serum. But
Yeah,
they could reliably detect it in both.
Yeah, I was looking at the RAP1B, for example, where you see more in serum than plasma, but it's not more, it's just a higher percentage. And so
That's correct.
It could be a threshold problem. Okay.
Yeah, and I guess there were also just 7 serum samples there.
Right.
So that's basically 6 out of 7 it was detected.
Yep.
Yeah.
Yep.
So can I ask a question about the difference between plasma and serum? I thought typically extracellular vesicles were being purified from plasma, not serum. Should we be looking at both populations?
Well,
I was just asking the question as expediency, and again, lots of clinical trials have collected serum and not plasma, and being able to go back to those. Is it stupid to use serum, or is it not unreasonable? That was my question, basically.
So yeah, I think it's a perennial debate in the EV field whether you should use plasma or serum if you have a choice. But sometimes you don't have that choice, as you note, and it really comes down to what your research question is. So if the major difference between the plasma and the serum is going to be that there's a lot of platelet generated products that you're going to find in serum and not in plasma. Now, on the other hand, if you don't deplete the platelets from your plasma before storage, then you're still going to have a lot of platelet material that is coming from the platelets after the blood is drawn. So it really does come down to what you're trying to ask and whether those platelet-derived particles are going to interfere with the answer to your question.
Yeah, they also show that there's a high overlap between the proteome of plasma and serum samples. So it might not be such a big deal. Like, it might not be a night or day thing, but yeah, there might be a clear preference for plasma for some experiments.
But this isn't yield. This is, or this is just whether the proteins are there or not, right?
This is the Pearson correlation. So if it's high in plasma, you would expect it to be high in CO.
Okay.
So we have several questions then. So Dan Lambert, Roger Alexander, Esther Naltatun, and then Ahmed also asks about what. So these are all having to do with this whole definition of EVs versus particles and where some of this material might be coming from. So I'd first just like to emphasize that the definition of the extracellular vesicle is something that is going to be naturally released from a cell. So it's not going to be the product of let's say something that would happen after you take the biofluid, and it is that bilayer membrane that defines the extracellular vesicle with cytoplasmic content. In contrast, an extracellular particle can be almost anything. So an EV is a particle, a lipoprotein is a particle. You also have various protein aggregates that can occur here. So Dan Lambert, did you want to jump in here, and then maybe Roger and Esther can also give their observations?
Yeah, thanks, Ken. Yeah, I'm just interested in the representation of extracellular matrix components in these preps, and how much of that material is picked up by vesicles traveling through the pericellular matrix, which might be exaggerated in vitro compared to what you might find in the circulation. So whether any of that material's sort of lost in the journey into the circulation. And I was getting the impression from some of the data that there might be some evidence for that here, but I don't know if anyone has any more insight into that, really, whether there's any evidence of an over-representation of ECM in the preps in vitro compared to in vivo.
Yeah, I wouldn't say from what I've looked at, I haven't seen an over-representation of the in vitro versus in vivo. Fibronectin, which is a very common extracellular matrix protein, is basically present in all of the samples regardless of source, which suggests that it's not specific to in vitro culture.
Yeah, I was interested in just very briefly in the Vesicle, which was pretty highly represented in a number of these data sets. It's perhaps something that could be picked up, particularly also, I guess, when you look at things like pancreatic tumors that are very desmoplastic, you'd expect to see a lot of things like Vesicle in that environment. So it might tell you a little bit more about tumor in terms of a biomarker.
Mm-hmm.
Any other comments on extracellular matrix proteins?
Yes, I was concerned about the huge amount of fibronectin, Ken, and Daniel, you two, we seem to be on the same page that this is not necessarily an indication that what we're seeing is associated with EV, or it could be, I mean, there's already fibronectin in the plasma to start with, and fibronectin increases upon inflammation. So, okay, but they've been very careful at defining this as particles rather than vesicles, so we can accept it.
Yeah, and it's interesting, what is going to be diagnostically most important. Those of us who think about purity of EVs and so on, we often, try to get as pure of a product as possible, and maybe that's important in some settings, but in other settings it might not be. Maybe it's better just to get this general particulate matter. So it'll be interesting to see how this can then translate, or how this can be verified in EV subsets that are derived from other separation methods.
Exactly. In fact, if you don't mind, Ken, stepping in, sorry that I didn't put the question there. Metastatic versus non-metastatic, for example, it would be very useful to know if there was we could differentiate between these two subsets. Did they mention if the patients were divided in the metastatic versus non-metastatic, just for what Ken is saying, for diagnostic purposes, or for whatever?
Oh, they did not. They only showed basically cancer versus non-cancer. They did not show different stages of cancer in this study.
Thank you.
Roger Alexander, did you have anything you wanted to add here about particles, EVs?
I guess I would just ask, I mean, it sounds like mass spec is definitely, you know, high quality data, and the ultracentrifugation is problematic. It'd be great to see them pipeline in new techniques into their mass spec pipeline. But since I sort of trust the data about the lack of finding the common surface markers we expect, and I wonder if you think that that finding is, you know, is strong and new biology, or what was your sense of it?
So I'm not super surprised about this, because basically these markers were identified from basically extracellular vesicles that have been isolated from cell lines. So because of the fact that we're not looking at extracellular vesicles that are actually in plasma and serum, it makes sense that we're not actually recapitulating that into our markers. What would be nice to do, though, is to take plasma and serum samples and do a much more rigorous isolation of the extracellular vesicles and make sure that this still holds up, that we're not just, because another way to interpret this data is that there's just so much other junk that's being co-precipitated in the plasma and serum sample that it's just diluting the signal out. So it'd be nice to do a much more rigorous isolation of the vesicles from plasma and serum to make sure that one, these proteins are not just the junk that's actually being commonly precipitated with the vesicles, but they're actually extracellular vesicle markers that are enriched in plasma and serum EVs that are not being captured when we just look at the cell line data.
Good. All right. So Esther, you had an interesting interpretation here. Can you share that with us?
Yeah, so my question was also not so much about whether things were associated to EV or anything else. I was a bit surprised to see, if I recall correctly, that in the cancer-specific profiles of the EVP, many of the proteins were actually immunoglobulins. And so the question is, what exactly would that mean? If they're really different between the different tumor types, if that is what I saw, yeah, could that mean that these are then anti-tumor antibodies, specific ones? Maybe then it becomes even more important also to compare the data to any immune-related diseases. So did they comment on this at all, or?
Yeah, I can't make any comments about the presence of immunoglobulins. This is the data set they used for identifying different types of cancers, and yes, you do see a lot of huge enrichments of immunoglobulins, which is very surprising.
Yeah, and this also I think goes along with another question that was asked from Kashipra Kapoor. Would the EVs extracted from plasma also contain EVs from healthy cells? And yes, they would. And we would not expect that all these markers are actually coming from the tumor. In fact, probably a minority of them would be. And so rather, we might be talking here about responses. And so indeed, it's interesting to see the immunoglobulins here and what that might mean for the interpretation of the study. So, so there's a question from Lilia, and I think it's quite interesting because maybe this data set can also be used to evaluate some of our models. So Lilia, can you bring up your point here, please?
Yes, thank you. So if I've seen it right, so they were showing that the correlation between the protein content from the cell lines, the mouse model, is low, is like absent comparison with the plasma and the serum. And they also checked, yeah, exactly this picture. And they also checked the commonly used the biomarkers for the vesicles, and they were not again present in the cell cultures. But then they found others, which were actually more correlated with their picture in the blood, in the serum, and in the plasma. So like those pictures that they have more candidates for in biomarkers. And my question is, whether they actually changed the cell model, or have they used the classical 2D cell culture, or that was something else? Because that might actually explain a little bit why we are so far away from the modeling of the cancer in vitro. So I'm just wondering whether there is any information about the cell lines and how this culture were actually done.
I believe all the experiments that were done in this paper were done in 2D cell culture models. So even for the tissue explants, the way they do that is they expand the tissue explants ex vivo, and then so it's a tiered culture, and then they collect the conditioned media and basically do the ultracentrifugation that they've done before. I don't believe they're doing any like spheroids or more physiological models.
So Lilia and others who are doing 3D culture, would you expect to see these kinds of differences?
Yes, indeed, because now we're actually working on the comparison of 2D culture versus 3D, and also the clinical situation, so the blood, and it's clearly that the 3D culture behaves way different than 2D in the protein content, RNA, and other things we're still investigating. So that's why I think that this picture for me looked like a challenge. And what shall we use then if the cell culture and the cell model and the mice model is actually not really correlating with the plasma? So I think that it will be also interesting to see maybe the study with a different cell culture in comparison with the plasma. Because we do believe that if the cancer actually grows in 3-dimensional, so it makes in 3D, that makes sense to grow that in 3D instead of the 2D, but that's again depends on the type of the cancer and the situation you're studying for. So yeah, that's why I've asked.
Mm-hmm.
Yeah, makes total sense.
Yeah, I think that's a very good point. So there's a lot I think that we still need to learn about how what we find in biofluids reflects what's happening in culture, and whether we can actually interpret some of our 2D cell culture results. Good. So Jay, you had a couple of questions here. I think starting with with protease. Do you want to elaborate on that?
Oh yeah, thanks, Ken. I think Roger answered the question. I'm trying to just kind of grapple with what's extracellular vesicle versus an extracellular particle. And one of my thoughts was that protease protection, at least for the contents within the lumen of vesicles, might be useful, but I realized that you'd lose the cell surface. I guess it's more of a philosophical question. I guess the sense I'm getting from you guys is that the particles, I mean, it's a very difficult biological question, but kind of the assumption is it's just kind of passively released junk. But at least in our studies of secretory autophagy, we have things that are not released in vesicles, but they are released as aggregates. And so they're biologically released in the sense that if you knock out genes in the pathway, they stop being released. So I guess, is there thoughts or any in the field with this kind of other stuff, these particles, whether some of these are biologically, or physiologically utilized or transferred, and also whether, they're being released based on, kind of different pathological states.
Mm-hmm.
Can I comment on that for a second?
Yeah.
Go ahead.
So I think there's two issues. One is with ultracentrifugation, there's a lot of junk. There's lots of extra things. Who knows what it is? But the other point is that, ribonucleoprotein particles in circulation, we just don't know enough. We can study EVs, but there's a lot of work left to do. We know Argonauts out there, but each little RNP, you have to have a specific way of isolating that, whereas EVs, there's sort of more, you can isolate entire classes of EVs and study them. So there's a lot of work left to do with RNPs.
Yeah, I think that's a great point.
Yeah, I agree completely. And, you know, we don't, I suppose if we want to put a really fine point on it, we should all just use the term extracellular particles, because we never can get a completely pure population of EVs. But we don't understand what all of that stuff is that we sometimes call junk. Maybe there's a lot of treasure in there too, that would be important for, not just for diagnosis, but also for understanding, coming back to Jay's point, whether this material is actively released and whether there are these biogenesis pathways or secretion pathways that are important for different particle types. So I think that's something that I see as an important part of the data set, the resource here that's shared with the community, that maybe we can get more information on some of those other particles that would then stimulate additional work. Let's see here. There's a lot of questions coming in today. We probably won't get to all of them, but we have a question about cryo-EM and whether we are going to be looking for anything in I guess based upon what has been reported here. Andy, are you using cryo-EM? I mean, would you be looking for what any of these things are?
Yeah, I'm not personally using cryo-EM. My impression is that the difference between cryo-EM and like transmission EM is that cryo-EM does a much better job of preserving the structure. One of the problems you have with regular EM is that you get desiccation of the sample. So you have a nice spherical EV before you process the sample, but then you you basically dehydrate the sample and the vesicle collapses, creating this cup that you can see on the right image, which we are now typically used to seeing for extracellular vesicles. And cryo-EM basically does a much better job of protecting the structure. Basically, cryo-EM with markers, immunogo labeling for markers of interest would tell you, though, if your marker is associated with the vesicle or not. But you can also get that data from regular EM.
Yeah,
I'm not an expert on cryo-EM.
Yeah, I think that cryo-EM it would give us a much better indication of what kind of heterogeneity of particles there is here. Sometimes, depending on the quality of the TEM, it's really hard to say for sure whether something is a vesicle or not. Camille, you have a technical question about MS.
Oh, hello. So I just wondering if when they were doing their mass spec, like showing figure 4, I guess, like how were they doing the quantification if they were running it at different times, if it's like label-free quantification?
I believe this is label-free quantification, but I'm not a mass spec expert, so sorry, I can't really address that question.
Yeah, I'm just curious because like if it's a tandem mass tag, like you can actually quantify, like do quantification easier when you have, even if you run it at different times, at least you can get that quantification. But with label free, I'm not sure how they're doing it. If you're normalizing it to a certain EV protein or like marker.
I'd have to go back and check how they're normalizing it, but I would guess that they're not normalizing versus specific EV marker, because that basically invalidate what they're doing up here, because they're basically saying that the EV markers are not really great. So it wouldn't make sense to me to try and use those markers to try and normalize your data when you're questioning.
Yeah. Yeah, it's also, I just want to add a comment that it's amazing how they did ultracentrifugation for almost like 500 samples. So.
Yeah.
Like how many people and how many hours were they spending doing ultracentrifugation?
Yeah, that's probably the reason why they didn't do like deeper methods like density gradients, because density gradients take an additional like 8 hours at least. So imagine how much time that would take.
Yeah, I think this was the culmination of several years, many years probably of work, just doing the regular centrifugation. So, well, and Camille, you also then made a comment about a 2019 paper comparing RNA profiles in 2D and 3D culture And indeed, there are several, I think the other questions that I'd like to get to and comments are bringing us back to this, this conversation about 2D versus 3D. And then, of course, it's not just that there's one kind of 3D. There's many, just as there's many kinds of 2D culture, you can also do a lot of different 3D kinds of models. So first of all, Camille, if, can you tell us something about that paper? I haven't opened your link.
Okay, so I haven't really looked at it in a while, and I was in one of these talks wherein the PI gave this talk. But one of the things that they were seeing, what there was a difference in like EV release alone, like in 3D culture, I think they were seeing that EVs were releasing in like 9 to 11 days, but in 2D culture, they were seeing EV release within 6 hours to 24 hours, and then by 48 hours, it levels off. But I don't exactly recall about the difference in the RNA profiles that they were showing, but most likely there is a difference.
Yeah, that
Yeah.
makes total sense. I mean, when you're growing cells in 2D, we're actually violating a lot of the common things that happen in vivo. I mean, we try to make sure that cells don't get too close together because it makes data hard to interpret. And but when you look at like tissues in vivo, all the cells are already touching each other. So you're going to have very different types of communication from cells that are touching each other versus cells that are not. So yeah, it makes total sense that you would see very different EV shedding patterns and also EV content from cells that are actually grown in a more natural structure as opposed to just being grown on an unnatural substrate such as plastic.
So what are, I think we have several people in here, John, Lilia, some others who are doing 3D culture. So what are these, what are the parameters that we should be thinking about with 3D culture? And so I'd ask John and Lilia perhaps to say a few words about that, and also to respond to Roger Alexander's additional questions about collecting enough material from these 3D cultures. How feasible really are these experiments?
I can. Oh, you can go first if that works.
Yeah, I can just say that it actually in our case depends on the cell lines. So some like fibrosarcoma cell lines, they don't actually, how I call it, they don't like 3D. So they don't produce that enough material per one matrix that you can work on. But in case of the prostate cancer, which I'm working on, they actually, some of the cell lines like the CBR and Nellicoms, they actually like 3D more. Because for instance, if somebody knows the Allentops cell line is very like tricky to culture it in 2D, they always die, they need some extra digits and so on. So, but it's actually not a problem because I can get the way higher, more particles per matrix in comparison with just a normal big plate. So because you also another point there in sort of hypoxia there, I think that also makes sense that there are many studies showing that hypoxia is actually increasing the particle production. And there is another part that the cells in, at least in our 3D cell model, you can grow them way longer than just in a normal 2D. They survive like for 7 days and makes sense. They produce more. They have less stressful factors outside. So I don't know, but that really depends because some of the cell lines, they don't actually like that. So they don't grow and they don't produce enough material, and we just go back to 2D, normal classical one. Yeah, if that's the answer.
John, do you have something to add?
Yeah, what we've found, and I mean, our group is not the only one doing it, evaluating ligand receptor interactions in 2D versus 3D culture has been going on for a while. But specific to tumor cells, in particular 231s, my group is, it's not my specific project, but I'm familiar with it. We've shown that you can induce dormancy by affecting both the stiffness and receptors available to the cells in terms of just bulk RGD that the cells can see in bulk MMP cleavable sequences, which makes sense intuitively, right? If cells have different ligands that they're seeing, it could certainly affect their behavior. And what I specifically have found with 231s is on collagen/gelatin versus fibronectin, they will grow and behave, I guess you could say, more aggressively faster than they would on bare tissue culture plastic. And while I haven't checked it specifically, kind of the running common thought is that the 231s are laying down their own fibronectin, collagen, et cetera, and that's causing a different set of processes to occur. And similarly, Shelley Payton's group has shown, I think it was a 2019 paper or maybe early 2020, that you can induce dormancy just by modulating the coatings available to the cells on the flask that you're using. And I think that that's important because anyone that's worked with 3D culture understands the difficulties it poses for producing cells in bulk, because you have to dissociate the matrix without affecting the cells to collect the EVs, which often bind to the matrix or just become sterically stuck in it. So I think what would be interesting for people moving forward to check is to look at where can you get away with coatings in 2D versus a full 3D model, if at all possible, or is 3D necessary for many cells, and how do we go from there? And of course, there's the final caveat of, it's going to be very dependent on cell lines. Some tumor cells are going to be much less sensitive to what they're grown on, and others are going to be much more sensitive, just the nature of where they are in the metastatic cascade and just cancer status as a whole.
Yeah, so a lot of factors to consider here. I think we've heard a lot today about this nice resource that's been made available, and how the conclusions of this paper, of course, must be couched against the realization that these are extracellular particles and not just EVs. And so perhaps some of these surprising findings, like the lack of CD63 and so on, that Catherine also brings up, are potentially due to the study design, at least in part. But a lot of provocative findings here, I think, and things that are going to be useful for the community in the years to come. So, all right, well, thanks everybody for joining, and a special thanks to Andy for being willing to present this paper to us. And I'd like to wish you all a great rest of the week, and hope to see you again soon. Take care, everyone. Bye now.
Thank you.